We'd like to thank Dr. Agustus for his excellent presentation. Now we're going to have Mr. Jorge López, President of the Colombian Foundation for Promotion and Development of Biorefineries from Valle University, Colombia, with his presentation, Biofuels from Natural Algae and Agro-Industrial Waste and the concept of biorefineries.
Good afternoon to everybody. I'm really trying this moment, bringing in this moment, the T-shirt of the first of the group in biofuel research and biorefineries concept. By the idea of biorefinery, we are in an Ibero-American network, which is now an Ibero-American Association for the Development of Biorefineries. We started basically under the part of the biofuels. We tried different types of materials, but the one that we have advanced the most is the one with the microalgae, and the other one which is residues of agro-industrial residues, especially the ones that are done through the sugar cane and the residues of cutting down. I want to make an emphasis, especially because this is the synthesis of what other people have said before, as to what is the concept of biorefining. But the biorefinery is trying to take advantage of a biomass where basically the biotechnology is what acts mostly to take advantage and value all the products that can be obtained from a biomass. And from there we get lots of products. We have a country that is biodiverse, and we are privileged because we are in the tropics, and in that sense we have a lot of possibilities to take advantage. Many of them, 90% of them, can be used as biocombustions, but as we saw in other presentations, there's a lot of compositions that can be taken out of the biomass, as much more than what we can get from petroleum. And that is a very important projection that we have been able to, we should consider in all the different things that we're going to do with this concept of biorefinery. Our group interacts with several disciplines. That's why it's an interdisciplinary, like it is in other groups, as they look for one specific application as to what is being done in the lab. Our principles are not to compete with food or the usage of land. That's an idea that we have. A lot of the materials that we are using are residues or products that are being used in the nature, or products that are be given in by nature, but it has not been used in other applications. The advantage of the biorefinery with which we work with in our group is that there are processes that are integral processes that everything that has to do with technology development has been become better, and for our company has become more rentable, more profit because of using the products, and it also the environmental impact has been reduced through our process as well, because we're using leftovers. And then also it's helping because we're having more jobs during this whole process of the biorefineries. And us especially, we're thinking on what us as a country in development we can do, because there's a lot of difficulties that we have, starting with resources, among other things. Here I would like to comment or make a synthesis of two of the parts that we are working on. We're using the residue of the sugar cane. We're using it the same as other materials that are lignocellulosic, like the trees. In that sense, the use of there's a big need to prepare the material that doesn't have much difficulties, but we always have to do it with pre-treatment to be able to make the extraction, for example, of the lignina, or it's one of the cellulosic materials, the proteins that must become hydrosoluble, which have to have essential oils. We have celluloses that can get also from because the cellulose is the chains of sugars that the 5 carbons and 6 carbons. Difference to the cellulose, which is basically of 6 carbons. So in this pre-treatment, we have been trying many methods. Within them, we find biological ones, enzymatic ones, chemical ones, and other things as well. And now we have found the process to be able to recuperate the lignina, the proteins, some of the hydrosolubles, and some of the cellulose. That pre-treatment basically is adapting the materials to make the hydrolysis happen and to make these polysaccharides into simple ones. The hydrolysis can also be done under biologic aspects or enzymatic aspects. And the idea sometimes, actually at this moment, is to do something that would be simultaneous of the hydrolysis and the fermentation. The fermentation at this moment, we did a prospectation, biological prospectation, in the northern part of the Valle and the coffee zone, to be able to get microorganisms, native microorganisms, because we have a problem here. The bacteria don't adapt easily from one place to the other. That problem we're living the sugarcane companies, because the bacteria they use from India are not adapting easily for them to be able to get the ethanol. And now through this process, then we're getting the distillation through which we can get specifically the ethanol or the xylitol, which is basically the sweetener for diabetes, people with diabetes. Okay, here we can some of the aspects that we have been able to find or we have been able to do within our group. I was already told you we were able to get some of these products. We have come to this point where we have finished the part where the lab, and now we're looking at the possibility of making a small a plant where we can start working on a bigger scale and working with sugar cane. And we have a difficulty because there's a residue of other cuttings that is very varied. So we want to do a plant that will process 2 tons of leaves and sugar cane. So now we're trying, we have this project trying to use the residue of the fuel, especially biodiesel and ethanol, because they're hydrolyzed from the sugar cane, or they can also be done from other materials, which is hydrocellulosic, to be able to produce butanol, which is a biodiesel, biofuel, that is much better than ethanol. And also using some processes of fermentation processes. Talking about microalgae, we have been able to use using the Phlerococcus and the Salidesmus. Here we've already talked about the good things that the microalgae has. There's enough information about this, and even there's annual congresses talking about this. In our case, We analyzed some of the characteristics of the composition, the characteristics and contents of the chlorella vulgaris. We have amino acids, carbohydrates. In that sense, we have focused essentially on the fatty acids, which look a lot like those fatty acids of other oil sources. In the case of the carbohydrates, we have more cellulose than in cellulose, contrary to what happens in the materials, in the hydrocellulosic materials. We did also a simulation based on the results that we have to do an evaluation of what a biorefinery would be. I'm not going to talk about the process here. I'm going to talk mostly about, first of all, the problems that we have in the treatment of the microalgae. Specifically, the cultivation of microalgae, it's very low. The production is very low, and that's one of our things that stop us. We barely get 1 gram per liter. So yes, the materials are not inexpensive. Well, at least in our medium, it's not going to be a rentable process. The other thing is the collection, the harvest of the microalgae. Since it's so low, it's very hard to be able to harvest. And there are some processes that are not easy, and we have tried to detail several methods. And in this sense, if they are inexpensive, it has been very in auto sedimentation, particularly by thanks to the change of pH. Another big thing is the disruption. The disruption is the breaking of the cellulose so that it will free the material what it has inside the cellulose, and this has been very hard. So then without that we haven't been able to do the lipid extraction. And one of those lipids, the most important ones are the phospholipids, and to be able to recover the protein. And all this to be able to do the biodiesel, but to be able to do this biodiesel, we need to refine the fatty acids, and that's where we are trying to decide if we should recover the proteins before or after, because that is a difficulty. Where do we recuperate? At what time do we recuperate each of the parts that are being able to get out? And at the end, what we finally get are the carbohydrates, and thanks to those carbohydrates we can produce ethanol. And like you have listened to before, we can also have the process, we can produce gas, and or what was left with is residues. The rest is to be able to produce the dyes, because dyes are very valuable. In our case, we have chlorophyll A, chlorophyll B, and among others. Here we can see some of the processes that we have been working on to be able to do the harvest. We have done photoreactors, closed, open, halfway open. This is the protein, this is part of what we have been able to do. This is another bioreactor that we also did. This is a technology that we, electromagnetic, so that we could do the disruption. Here we can see initially how the harvest comes out and how we have to concentrate it, and here it's more or less concentrated, sedimented. But then that paste has a very low concentration of biomass. The concentration of water is approximately 90% more, but it still ends up looking like a paste. Another thing that we have tried a lot is the part where we try to get the information and to control some of the parameters which are important, like the pH, the oxygen, which is terrible for the microalgae. Oxygen is a problem, and they consume CO2. These we have already automized it in our pilot plant. doing cards and doing automated signing the feeding and the process of that happens after the harvest. I would like to talk a little bit about how our foundation was born. We consider in recovery through the country that there are a lot of things that are repeating. There's no synergy. Each university, each group of investigation is oriented, let's say, for something that is important, but it might not be the most relevant, or maybe not what the country needs the most. So from there, the idea to have an organization that we called Biorefinery, which is the promotion of the refinery, to defend the development that is being done in as a group. So then we established different seatings in different parts of the country, and the idea is to be able to make a call, especially to the entities, especially that could be interested in our foundation, because it's the only way for us to have strength. Initially, we have representatives of different entities, but we still haven't been able that the entities get integrated as members of our foundation, and without that we cannot obtain this objective, which is to establish the national policies and to be able to work together, that investigation, this basic investigation, and to be able to apply it into the productive chain in the use of the biomass in Colombia. And I hope that through this we can talk with this in this program and share with you all. Thank you very much for this invitation, and thank you for giving me this space to talk.